My name is Jinjie Liu, and I'm a fixed-term assistant professor at Michigan State University. I'm from China. At the mainland, I got my PhD from Ocean University of China in molecular genetics. In 2018, Christoph Benning came to me, said, "Hey, we have this cool thing. Do you want to do it?" QR stands for Course-Based Undergraduate Research Experience. So basically, it's a pedagogical approach that integrates research projects into curriculum. So the students in the classroom would do a research project. I went to talk with John Stosfos from Biological Science program because he is the part of the initiation for this CURE project. We started collaboration. He has provided tremendous amount of support. You know, for somebody who had never taught at US, I was a little bit timid, but I really want to do this. I have been really wanting to work with students, basically do research. That was my dream job even when I was in college. The research project is developed from big NSF-funded project, lipid signaling involving chloroplast implant. The students in the classroom only works on part of the project. We are trying to provide authentic research project experience in the classroom. Basically, students start asking the research questions, formulate hypotheses, and then they design experiment, and all the way down to data analysis, and then later they start the communication, like the poster session at the end of the semester. At the beginning of the semester, the students sow the seeds, and then while the seeds, they wait for the seeds to germinate, we wait for the plants to develop. So we have some pre-prepared materials to train them the skills, how to observe the phenotype, what measures they are going to take. When the seeds germinate, when the plants are about maybe 4 weeks, 5 weeks old, then the students start picking the mutants based on their research hypotheses. They are going to take measurements. They compare these mutants with their controls. That is for the phenotype part. And of course, when it's come to the genotype part, they see because this is mutants, the genetic background is different. Then they compare that with their control to see whether this is a real mutant or not. Once you identify the mutants, right, once you check their genetic background, there's tons of work you need to do. We cannot do that in the classroom settings, so we moved to the research lab. And at that time, the research lab provided the DNA sequencing data to the classroom. So the students start mining the data there, looking for different types of mutations. We have several students who have worked with us after they finished the CURE session. Yeah, so some students, once they took the course, they got really interested in research. There's reports saying that the CURE, like having students involved in research that help them in many different ways. They have better understanding about course content, they develop better critical thinking skills, so better plan their future careers. On top of that layer, I want them to get a taste of what scientists do, how to ask questions, how to design experiments, how to collect data, and how to support your claim. These skills are transferable. We offer this opportunity to students. They can make a better judgment whether they want to pursue a STEM career or they do not, because many students probably won't think about doing research. But for me, I think running this course for 4 or 5 years now, it's the excitement I was seeing from the students. They like this opportunity and they report very positively. For the data we collected by running this course, the students have very good perception about the scientific process, how they ask the questions, how they use data to support their claim, how they analyze the data, and how they draw the conclusions, how they become a part of the community. I have enjoyed working with them, but their positive feedback certainly is very encouraging for me to continue improve the course.